Okay, so welcome everyone to the Division of Rheumatology Grand Rounds. Before we get started, just a couple reminders that Dr. Jonsson is going to save about 10 to 15 minutes at the end of her lecture to answer your questions. So please feel free to type your questions into the Q&A box throughout the lecture. And also, if anybody at the end would prefer to ask their question over audio, you can always raise your hand and I can promote you and give you that ability to ask your question. And this session will be recorded and posted on our website as well. So now I'm going to pass things off to Dr. Mustelin.
Okay, thanks, Lara. So good morning, everybody. It is a great pleasure and honor today to introduce a fellow Scandinavian, Dr. Helena Jonsson, who received her PhD and MD degrees both from Washington University in St. Louis, 2010 and 12, and then did a internal medicine residency at Brigham and Women's Hospital in Boston, and continued on there with a rheumatology fellowship, and is still at Brigham and Women's Hospital now as an associate physician and very much a physician scientist. Her interests in rheumatology started early on. Her PhD work was on natural killer cells in autoimmunity, and she has focused very much her research career on human immunology and trying to understand what role particularly T cells and CD8 T cells in very much in diseases like rheumatoid arthritis and lupus. And she has a very active role in the Accelerating Medicines Partnership, which probably most of you know about. It's an NIH-supported large collaborative effort to profile in depth the immune system in both tissue and blood of patients with RA and SLE. And today, Dr. Jonsson will talk to us about her work with the CD8 T cells in autoimmunity, and particularly the Granzyme K expressing ones, a very interesting, recently discovered subset. So I'm just going to add that Dr. Jonsson has published some really impactful papers in Nature Immunology, Nature Medicine, Nature Genetics, and many other high-profile journals. And I'm going to give the rest of the time to her to tell us about her interesting research. So please, Dr. Jonsson, go ahead.
All right. Well, thank you so much for that introduction and for the opportunity to speak today. I'm really excited to tell you about my research, and I definitely look forward to all your questions. So please, I wish you could interrupt me in the middle of whatever I'm saying, but please do type them into the Q&A, and I'll get to them at the end. So I focus mostly on rheumatoid arthritis, but the cell type that I look at turns out to be important in a number of different diseases. So I'll touch on that a little bit throughout the talk and then again at the end. But most of this talk will be about rheumatoid arthritis, which everyone is familiar with. And so I won't go into further detail about that. But what I will mention about rheumatoid arthritis is that it's pretty obvious to the clinicians that the site of disease most commonly is the synovium, right? It's the tissue. And that's where we as researchers also need to focus. A lot of studies, especially in human translational research, focus on blood. But I think what the project I've been working on really shows is that the key is to find the cells that are responsible in the tissue. So here I'm showing two images. On the left is a microscopy of normal synovium. This is at just H&E stain, and you can see lots of connective tissue fibers and a few cells here in the sublining, and then the lining layer is very thin, consisting of some macrophages and some fibroblasts, and those are the main cells that are present in normal synovium. But in rheumatoid arthritis, the synovium is enormous, mostly because of this influx of leukocytes. So there are neutrophils, there are monocytes and macrophages, there are different types of T cells and B cells and other cell types all converging. And in addition to that, there's expansion of the fibroblast populations and of the resident macrophage population. So everything coming together in a very complex picture. And this is the type of thing that we're just now sort of starting to understand and put together. So when I was learning about rheumatoid arthritis, in medical school and during fellowship and so forth, these are the sort of classic images that were always shown, especially this one here in the middle, the sort of cartoon inflamed knee that we always see, where one half is normal and, doesn't really have a lot of cells going on. And then the other side reflects the inflamed knee and sort of lists the cells that I just counted off. And T cells are always on that list. But then when you look at the more detailed images here and here, it's always CD4 T cells, right? It's always CD4 T cells that are mentioned, so in an image like this is from that same review from Lancet in 2016, for example, we have Th1 cells, Th17 cells, regulatory T cells, all the different T cell types that are mentioned are all CD4 T cells. But what about CD8 T cells? What are they doing? Are they not involved? Are they not important? Nobody really knew. There'd been some work done, but not a whole lot. And I mean, there are good reasons for that. So the focus in rheumatoid arthritis, it's been on CD4 T cells for several reasons. One, in genome-wide association studies, the HLA class 2 genes are, by far the strongest signal, pointing to a CD4 T cell class 2 interaction driving this disease. CD4 T cells are also the classic cytokine-producing T cell population, whereas CD8 T cells are considered to be killer T cells, and maybe that doesn't make as much sense in rheumatoid arthritis. And then, of course, CD4 T cells are able to provide B cell help, to allow B cells to make autoantibodies like the autoantibodies we see in rheumatoid arthritis. And so, all of this came together in a very pretty package. Obviously CD4 T cells, very important in rheumatoid arthritis. But is that the whole story? It turns out that CD8 T cells are just as frequent as CD4 T cells in synovial tissue. So this is looking at osteoarthritis and rheumatoid arthritis synovial tissue that's been disaggregated and then stained for different cell types. And here we're looking at the frequency of CD4 T cells on the left and CD8 T cells on the right among total live cells. So among fibroblasts and neutrophils and everything that's in that tissue. And it turns out that CD4 T cells, usually span somewhere on roughly between, 10 and 30% of live cells in inflamed RA synovium. And the exact same is true of CD8 T cells. So CD8 T cells are present in just as high a number as CD4 T cells. And when you look among T cells to see what fraction of T cells in synovial fluid and synovial tissue are CD8 T cells compared to CD4 T cells, it actually turns out that CD8 T cells are enriched. So in RA blood, for example, about 20% of T cells are CD8 T cells, but in synovial fluid, that fraction is doubled. So about 40% of T cells in synovial fluid are CD8 T cells, and maybe a little less, like 30 or 35% in synovial tissue. So in other words, there's a selective enrichment of CD8 T cells going on in both synovial fluid and synovial tissue. So let's take a step back and sort of review, what these two cell types do. So I touched on this a little bit before, but CD4 T cells, so CD4 T cells bind to class 2 molecules, such as HLA-DR, which again are a big deal in rheumatoid arthritis, according to these genome-wide association studies. They make a lot of cytokines, and cytokines are how the immune system communicates. And so these cells, when I'm explaining this to patients and so forth, I sometimes call them the CEO of the immune response. They're sort of taking signals and responding with other signals and sort of really directing what type of immune response is formed. And in accordance with that, there are also many different types of populations. In rheumatoid arthritis, we think about peripheral helper T cells, regulatory T cells, Th1, maybe Th17. All these different cell types are thought to have some sort of role in rheumatoid arthritis, either in animal models or in humans. So what about CD8 T cells? The stereotypic view of CD8 T cells is that they kill target cells. So, virally infected cells and cancer cells, that's why there's a lot of emphasis on CD8 T cells and tumor immunology, for example. And so these cells are more like assassin worker bees. And, what is an assassin worker bee going to be doing in rheumatoid arthritis synovium? So let's look a little more closely at that, though. Like what makes up these cytotoxic T lymphocytes? So CD8 T cells, they have a T cell receptor, that's very similar to a CD4 T cell, except this T cell receptor binds to MHC class 1. So this is the HLA molecule that's found on all cells in the whole entire body, not just antigen presenting cells. The typical effector cell that's a CD8 T cell is often called a cytotoxic T lymphocyte, or CTL. And what CTLs do is that when they bind to MHC class 1, they release a whole bunch of cytotoxic molecules. So those cytotoxic molecules include granzyme B, which we're going to come back to. They include perforin, which is a molecule that aggregates into pores in the membrane of these target cells, and granulysin. And these different proteins come together. The perforin again forms a pore, allowing the granzyme B and granulysin to enter target cells. And inside the target cell, granzyme B, granulysin, and other cytotoxic proteins, they cleave caspases to elicit apoptotic caspase cascades and other proteins, ultimately leading to death of the target cell. And that makes a lot of sense in viral infections, in tumor immunity, that sort of thing. But it might not make much sense in rheumatoid arthritis. So again, this is the classic molecule for cytotoxic T lymphocytes is granzyme B. So I want you to keep that in mind as we move forward here. So let's see what CD8 T cells in rheumatoid arthritis might be doing. It turns out actually that CD8 T cells make a lot of cytokines. So when we looked at CD4 T cells and CD8 T cells, again, these are from synovial fluid, and we just look at how many, what level of cytokines these cells are making. We actually find that CD8 T cells make more interferon gamma, so more CD8 T cells are making interferon gamma than among the CD4 T cells. And when we look at TNF, which of course is a classic rheumatoid arthritis cytokine, CD8 T cells make it at a slightly lower frequency than CD4 T cells, but still at very respectable levels, with almost all the cells responding when you stimulate them by producing TNF. When we look at the total pool of T cells making either interferon gamma here on the left or TNF on the right, we see that CD8 T cells form more than half of the population of T cells making interferon gamma after either T cell mediated stimulation here or a non-specific chemical stimulation on the right. And for TNF, they're a slightly smaller fraction, about 40% of T cells making TNF are CD8 T cells. So in other words, while the CD4 T cells have been getting the credit in most of the RA literature, CD8 T cells are just as potent as making these cytokines. And from what we've found, they're probably making about roughly half of the interferon gamma and TNF seen in RA synovium. So with the advent of single-cell RNA sequencing, we got what was maybe the first really unbiased look at CD8 T cell populations in rheumatoid arthritis. And those findings took us by surprise. So here I'm showing single-cell RNA sequencing from synovial tissue, showing T cells from about 25 different synovial tissue samples from patients with rheumatoid arthritis. As I showed you before, CD4 T cells are about half of the synovial tissue T cells, and CD8 T cells are about the other half. The CD8 T cells here are in green and blue. The CD4 T cells contain the expected populations like these TPH cell cluster up here, which also includes a few Tregs. But among the CD8 T cells, the clustering actually surprised us quite a bit because most of the CD8 T cells fall into this large green cluster, which is marked by very high expression of granzyme K, but low expression of granzyme B and low expression of perforin. Only a small number of CD8 T cells here in blue express that typical cytotoxic T lymphocyte pattern of high granzyme B and high perforin. And so this took us by surprise. What are these CD8 T cells that are forming the majority of the population? Because they don't look like the effector cells that we're used to seeing. The first thing we did is we looked by histology to see if in non-manipulated tissue that hasn't been disaggregated and sort of really messed with in a lot of ways, do we still see these CD8 T cells that express both granzyme B and granzyme K? And it turns out that we do. So these cells marked with the arrowheads here, these cells express CD3 in green, granzyme K in red, and granzyme B in yellow. And so you can see that even in this tiny little segment of an inflamed RA synovium, you see a number of T cells here that express both granzyme K and granzyme B. And so these double expressing cells, they're definitely present. We next used flow cytometry to quantify these different populations. So here, these flow cytometry plots are gated on CD8 positive T cells from RA synovial fluid, RA synovial tissue, or healthy control blood. On the x-axis, we see staining for granzyme K, and on the y-axis, we're staining for granzyme B. And what you see is that in synovial fluid and synovial tissue, the majority of CD8 T cells express both of these molecules, whereas only a small number express granzyme B alone without granzyme K. And that's drastically different to healthy controlled blood, where most of the CD8 T cells are naive and express neither granzyme, and very few express both together. Instead, you see some cells that express only granzyme B and some cells that express only granzyme K. So here we're seeing, a distinct population of cells present in RA synovial fluid and synovial tissue that expresses granzyme K and granzyme B together. And when we look across a number of different samples from these different tissue sources, we see that pattern play out, where the granzyme K, granzyme B positive CD8 T cell population is uniformly the largest in synovial fluid. In synovial tissue, it's a little bit more even, but in both settings, this granzyme B positive population that looks like a CTL is very rare, with only maybe 5% of cells in both synovial fluid and synovial tissue. And that's drastically different from healthy controlled blood here on the right, where most cells are naive and express neither granzyme, and the granzyme K positive, B positive population that we see so abundantly in synovial fluid and synovial tissue is very rare, maybe 10% on average. Now, I know many of you study lupus, and I want to point out that this population is also seen in lupus nephritis. So again, a tissue site affected by lupus. This is from the AMP consortium paper about lupus that was published in Nature Immunology in 2019. This is the single cell RNA sequencing data showing most of the leukocyte populations present in rheumatoid, or in lupus nephritis. And you see the clusters are called CT for T cells, D for dendritic cells, M for macrophages, and so forth. So you see there are a number of different T cell clusters. And it turns out that one of them here, the CT4, is a granzyme K positive CD8 T cell cluster with gene expression very similar to that same cluster that we saw in rheumatoid arthritis. Here, it's only one rather small cluster out of many T cells clusters. And so there's something a little bit different going on in lupus nephritis that I'll touch on again later, but we see it again in inflamed tissue. So what is granzyme K, and what do we know about it? So granzyme K is a protease, just like granzyme B, meaning it can cleave peptides, but it actually comes from a different family of proteases. So granzyme B cleaves caspases and other sort of cysteine expressing proteins, whereas granzyme K is more closely related to trypsin, which is something we all use in tissue culture and so forth. Granzyme K, in other words, cuts at different moieties in the proteins. And so it cuts a different array of proteins compared to granzyme B. The literature on granzyme K is not very big, so we don't know a ton about what it does, but the expression pattern of granzyme K has been documented during the course of CD8 T cell development. And that's what I've made a cartoon to show here. So CD8 T cells leave the thymus as naive CD8 T cells. Somewhere along the way, they encounter antigen and become activated. And it's thought that they sort of move through several different kinds of stages before they become an effector cell. And those are often called early memory, late memory, and then effector T cells, which for CD8 T cells are often called TEMRAs, so T effector memory that have re-expressed CD45RA. No need to remember that. That's not going to come back in the talk today. But the pattern of granzyme K expression that's been described is that granzyme K appears after the cell has been activated by antigen, and then it's present in these memory populations, but it goes away at the same time that granzyme B expression appears. And so most of these studies were done in healthy controlled blood. And this, again, correlates pretty well with what we saw in blood, where we saw some cells that express granzyme K alone and some cells that express granzyme B alone. But this type of picture doesn't really give us a good idea about why we see such an accumulation of these double expressing cells. Why are we seeing an accumulation of cells that would, according to this developmental pathway, sort of wind up in this transitional state? Cells don't often get stuck in a transitional state. So what's going on here? And what we're proposing is that this is actually a new sort of terminal CD8 T cell effector type that people have not described before. So why do we think that? So let's talk about the different roles that these cells are likely to have in RA synovium. So the first thing we wanted to look at is cytotoxicity. So cytotoxicity, again, is the hallmark of classic CD8 T cell effector function. Are these cells cytotoxic? And it turns out that they have very low cytotoxic potential. So when we look at the expression level of granzyme B and perforin, these two classic cytotoxic molecules that are made by effector CD8 T cells, we see that they're very high in granzyme B expressing cells in blood. That's this blue bar here, healthy control blood expressing granzyme B. And for perforin, we see that level here. What we see here is that the granzyme K positive, granzyme B positive populations and the green bars have about half the level of granzyme B as these typical CTLs. And that's true both in healthy control blood and in the RA synovial fluid. So we think that these granzyme K positive, granzyme B positive CD8 T cells are not cytotoxic, that they have other effector functions. One thing that came up pretty early is, are these CD8 T cells exhausted? So a lot of the CD8 T cell literature circled around tumor immunology, as I mentioned before. And in tumor immunology, there's a big concern that CD8 T cells that are present in the tumor have become sort of inert or exhausted, and therefore they're not helping to fight off the cancer. And so one thought was that, maybe these CD8 T cells are exhausted, but we don't think that that's the case. And these are the reasons. So one, there are several activation markers that are expressed by CD8 T cells that tend to be expressed by CD8 T cells that are actually doing things as opposed to, sitting around being exhausted. And one of those is HLA-DR. And here you can see that in RA synovial fluid, the granzyme K and B double expressing CD8 T cells and the granzyme B positive CD8 T cells express these two, so HLA-DR and CD69, at roughly the same levels. It's not like one population has very low activation markers and the other one has high ones. So we think that these cells are activated similar to other cells. What about proliferation? So these granzyme K, granzyme B positive CD8 T cells, they do seem to proliferate a little bit less than the granzyme B positive CD8 T cells. And we have some thoughts about why that is, but it's something we're still looking at. But when we look at the markers of exhaustion, so these classic markers of exhaustion from the tumor immunology world, like TIM3, ICOS, PD1, and LILRB1, we see that these markers are not expressed at higher levels on the granzyme KB double positive CD8 T cells compared to the granzyme B. If anything, the granzyme B positive CD8 T cells express more. And so we do not think that this is a specifically exhausted T cell population. And lastly, the reason we don't think that this cell is exhausted is that when we stimulate these cells, they respond just as well as other CD8 T cell populations. So here, looking at synovial fluid, we see that the green and the blue bars are roughly the same in terms of interferon gamma expression and TNF expression after a non-specific chemical T cell activator, or after TCR-mediated stimulation with anti-CD3 and CD28 beads. So we think that they're just as functionally potent as other CD8 T cell populations. So, so their activation seems the same. They make TNF, they make interferon gamma, similar to other CD8 T cell subsets in RA synovial fluid. What's special about these cells? So obviously one thing that's special is that they make granzyme K. So what does granzyme K itself do as a molecule? We talked before about how it doesn't cleave caspases. Like, what else might it be doing? And again, this is a very small literature, so we're kind of in uncharted territory here. And so that's why we decided to start looking at this using an in vitro system with synovial fibroblasts. And the reason we picked synovial fibroblasts is that synovial fibroblasts are present both in normal synovium and in RA synovium. So even here, this histology is sort of dominated by these leukocytes that have infiltrated it. Around everything, we have these RA synovial fibroblasts. And we also know that RA synovial fibroblasts are different from the ones in healthy synovium. They've been reprogrammed to make inflammatory cytokines like IL-6 and so forth. And we were wondering if granzyme K might have a role to play in that inflammatory reprogramming. And what we found, so the first thing we did was to test to see, can granzyme K kill RA synovial fibroblasts at a range of concentrations? And our results show that no, granzyme K does not kill synovial fibroblasts. either when you add it extracellularly. So here we have a range of concentrations of granzyme K added to the culture medium. But even when you use a protein transfection reagent to put the granzyme K into the synovial fibroblasts, you still don't see an increase in cell death. And that's shown here with various ranges of granzyme K put into a protein transfection reagent and transfected into synovial fibroblasts. So again, we don't see any evidence that granzyme K induces cell death. Instead, we find that that CD8 T cells, that granzyme K can induce synovial fibroblasts to produce IL-6, again, this classic pro-inflammatory cytokine that we all know about, and also chemokines such as CCL2, which help to recruit other cells. And here you see a dose response curve for both of them, where we see increased expression of IL-6 and CCL2 with exposure to recombinant granzyme K. The other thing that we found more recently is that granzyme K seems to be able to work together with interferon gamma and with TNF to amplify these responses. So here, for example, we have IL-6. We see that granzyme K alone can induce some IL-6, as can interferon gamma alone. But if you add them together, the response is bigger. Here it looks additive. In other experiments, it looks a little synergistic. It's a little bit variable. But either way, you know, the response is amplified. So it really seems to be true for IL-6, a little bit less so for CCL2, where granzyme K has a relatively small effect, interferon gamma has a bigger effect, and if you add the two, you see the granzyme K providing a tiny little boost to what interferon gamma can do alone. And here I'm showing the effects of TNF. So with interferon gamma, or with IL-6 rather, we don't see much effect with granzyme K alone in this particular experiment, nor with a low dose of TNF. But if you add them together, you get a rather hefty IL-6 response, similar to if you had 10-fold higher doses of TNF alone. And that's again seen for IL-6, whereas with CCL2 production, we don't see the same effect. We see granzyme K inducing some level of CCL2, we see TNF inducing some level of CCL2, and then the two together, it's a little bit higher than each one individually, but it doesn't even reach the additive stage. So one other question that comes up in the CD8 T cell field, and that's becoming more common in the it's something that's become a question in the rheumatoid arthritis field too, is this question of resident memory T cells. So resident memory T cells, this is a field that started out actually in dermatology, where dermatologists noted that particular rashes, such as contact dermatitis and psoriasis, these rashes tend to appear in the exact same spot every time somebody flares. So if they have a patch of psoriasis on their right forearm, for example, every time their psoriasis flares, that particular region of their forearm forms a plaque. And it turns out that what drives that seems to be that some CD8 T cells, called resident memory CD8 T cells, they stay in the skin there. And they stay forever, they never move. And every time a flare happens, those resident T cells help to elicit that flare to occur. And there's been a thought that maybe that explains why rheumatoid arthritis tends to flare in the same joints as well. Maybe there are resident memory T cells in synovium. So one question about these CD8 T cells that we saw was, are they resident memory T cells? This is a bit of a, it's a relatively new field. The markers of these cells are a bit in flux. How to define them is a bit in flux. But we tried to look at this using bulk RNA sequencing. So we sorted cells that were stained for granzyme K and granzyme B, and then performed bulk RNA sequencing on these cells and looked at, in this case, 4 different markers of T resident memory cells. And these granzyme K, granzyme B positive CD8 T cells are shown here in green, as they are on all the other slides. And then the granzyme B positive cells are shown in blue. And you can see that in each case here, the green expression level of each of these markers is either lower or the same as the granzyme B. So they express lower levels of CD103 and Hobbit, and about the same level of BLIMP1, which is another transcription factor for resident memory T cells, and about the same level of BRNX3. So we don't think that these are T resident memory cells. So if they're not T resident memory cells, where are they coming from? And how do they turn into what they turn into, these granzyme K positive, granzyme B positive CD8 T cells? And this is something that we're still working on. We're sort of making progress bit by bit. There are various ways to look at that, but it's very difficult with human beings because you can't, get synovial biopsies, you know, on a even an annual basis or something like that, that's a little too invasive. We're working on setting up mouse models to look at this eventually. But right now we're trying to use next generation sequencing technologies to figure this out. And so one thing we've done is TCR repertoire analysis of matched blood and synovial fluid. So our theory is that TCR repertoire analysis gives you the T cell receptor sequence of each individual CD8 T cell. CD8 T cells that come from the same progenitor, you know, so sibling CD8 T cells with the exact same T cell receptor, you know, they all evolved together. They differentiated from a single source. And so if you look at sister clones in the blood and in the synovial fluid and then compare what their phenotypes are, that can help you figure out, you know, how one cell became another. You know, did they develop in the blood? Did they differentiate in the synovial fluid and so forth? And so here I'm showing one of our early experiments. We have matched blood and synovial fluid from 2 patients. One patient is here on the top and the second patient is here on the bottom. We sorted CD8 T cells and then obtained TCR repertoire analysis and single cell RNA sequencing analysis from the same cells, you know, CD8 T cells from all of these individuals. And then we pooled all of those and analyzed them together. So this is single cell RNA sequencing where each dot is one cell. Here where I'm showing granzyme B expression as a heat map. So you can see granzyme B positive CD8 T cells are mostly here in the lower left corner with a little bit up in the upper left and a little bit in the lower right. Granzyme K expressing cells tend to be located up here, sort of on the top right portion with a little bit of expression in the bottom right and top left. So when we look at these sister clones, you know, these clones that are present in both blood and synovial fluid, our question was, you know, are they in the same spot? You know, do they, if they localize to the same spot on this map here, in each case, that means that the blood and the synovial fluid cells are phenotypically similar, which would suggest that they differentiate somewhere else and then migrate into the synovial fluid and tissue as sort of ready-made cells. Whereas if the blood sister clones looked one way and the synovial fluid clones looked a totally different way, that would suggest that cells arrive and then differentiate inside the synovium and the synovial fluid into this unique granzyme K positive, granzyme B positive phenotype. And what we found is that, you know, so here this in dark blue and in light blue, we're marking the top 3 sister clonotypes for patient 1, and then the top 3 sister clonotypes for patient 2. And you can see that the blood sister clones and the synovial sister clones are essentially in the same spot here. So for patient 1, it turns out to be this bottom right corner, which happens to be an area that expresses both granzyme K and granzyme B. So that suggests to us that these cells that are really enriched in synovial fluid, they probably came from a granzyme K positive, granzyme B positive pool in the blood, even though that pool is very tiny. And we see a similar thing in patient 2. So here, the blood sister clones are in a little bit of a different area on the map, right here with high granzyme K expression and low granzyme B expression. And when we look at the sister clones in synovial fluid, we see that they're mainly in that same general area with a little bit of spread here. But that again tells us that these cells, you know, either they're differentiating in blood and then arriving in the synovial fluid as ready-made cells, or alternatively, that they actually might be migrating into and out of the inflamed joints. So you might have this sort of recirculation phenomena going on where blood and synovial fluid pools of CD8 T cells are coming back and forth. And it's specifically these granzyme K positive CD8 T cells that do this migration. And so that, you know, with these data, we've drawn up this model of how we think that these cells work. So we think that granzyme K positive, granzyme B positive CD8 T cells are the ones that migrate in from the synovial fluid. And that's based on repertoire data that I just showed you, and also based on chemokine receptor expression that I have not shown. In the RA synovium, they receive signals maybe through their TCR, but maybe also through cytokine receptors, because it turns out these cells can be activated by cytokines independent of antigen. And they respond to these stimuli by making lots of interferon gamma, but also TNF and other things like CCL5, and also by releasing granzyme K. Granzyme K targets other cells in the synovium, such as synovial fibroblasts. And in response, the synovial fibroblast makes IL-6 and CCL2, and also in other data that I didn't show, reactive oxygen species, which is another compound that we know is enriched in inflamed RA synovium. And the granzyme K can interact with interferon gamma or TNF to amplify these inflammatory responses. So this is how we think that it works in rheumatoid arthritis synovium. Our next question was, are these similar granzyme K positive CD8 T cells also seen in other inflamed tissues? And one of the reasons we thought about this is, we'd seen that lupus data that I showed before from 2019, where one of the clusters was granzyme K positive CD8 T cells. We wanted to use next generation sequencing to look more broadly at more diseases to see if there were signs that these granzyme K positive CD8 T cells with this low cytotoxicity type of phenotype were present in other types of tissues as well. And so the way that we did that is we went back to our RA synovial tissue single cell RNA sequencing data set that I showed at the very beginning of the presentation. And we created gene scores for the granzyme K positive CD8 T cells and the granzyme B positive CD8 T cells, so that we could easily look at other RNA sequencing data sets to see if the granzyme K positive CD8 T cell score predominated or the granzyme B positive CD8 T cell score. So, as you might expect in RA synovium, we see high expression of the granzyme K positive CD8 T cell score and low expression of the granzyme B score, and that correlates with, both the protein and RNA data that I've already shown. We were a little surprised to see that even in osteoarthritis synovium, which has a very small number of CD8 T cells, but they are there, we still see a predominance of the granzyme K score. So here in synovial tissue, both inflamed and non-inflamed, or at least less inflamed synovial tissue have a high, a high signature for these granzyme K positive CD8 T cells. Next, we looked at publicly available RNA sequencing data sets from Crohn's disease, both inflamed ileum and non-inflamed ileum from patients with Crohn's disease, and also in an ulcerative colitis data set consisting of, these are again intestinal biopsies from inflamed colon from a patient with patients with ulcerative colitis, or biopsies from non-inflamed portions of the colon from patients with ulcerative colitis, compared to biopsies of healthy colon. And in all of these cases, so in all five kinds of intestinal biopsies from both inflamed and non-inflamed sources, the granzyme K positive signature predominates, whereas the granzyme B signature is very low. We next looked at lupus nephritis. So we had a couple of lupus nephritis data sets that we looked at compared to biopsies of healthy kidney. And this is actually where the pattern looks a little different. So in lupus nephritis, we see strong granzyme K scores and granzyme B scores. So, we're still seeing a strong signature for granzyme K, but there's something a little different about lupus nephritis, where the CD8 T cell populations are in a different sort of balance than what we see in intestine or in synovium. And interestingly, healthy kidney looks a lot more like osteoarthritis, for example, or healthy colon, with a predominance of granzyme K CD8 T cells. And of course, this being the pandemic, we wanted to look at an infectious setting. So we looked at a bronchoalveolar lavage single cell RNA-seq data set from patients with severe COVID-19, mild COVID-19, and healthy lung. And here as well, in the severe COVID-19 bronchoalveolar lavage fluid, we see that the granzyme K signature is a little bit stronger than the granzyme B signature, which was a surprise to me, because I had thought that, in the context of an acute viral infection, you would see a strong granzyme B score. But here we see that even in infectious disease, tissue seems to have a strong granzyme K score and a lower granzyme B score. So in summary, I've shown you about granzyme K positive CD8 T cells, and it looks like that they're the dominant CD8 T cell state, not only in rheumatoid arthritis, but according to the single cell RNA sequencing analysis that we've done, it looks like they're also present in a number of other diseases and also in healthy tissues. And so this is a CD8 T cell subset that we need to learn more about, because they seem to likely to be playing a role in a number of different disease contexts. Granzyme K does not drive cytotoxicity, but it activates target cells such as synovial fibroblasts to produce pro-inflammatory cytokines and chemokines. And it's likely doing the same in other disease settings. And so, we need more literature about Granzyme K. We shouldn't assume that all granzymes work in the same way. And there are a number of people I want to think about for this project. So I work in the lab of Michael Brenner here at Brigham and Women's Hospital, who has been an excellent mentor. And a number of people in the lab have had a role in this project. And the entire division as well, since all of the work I've shown you is human immunology, and we rely on patient samples. So many thanks to the patients and also to the clinicians who've helped recruit them to our biorepository. This project is a partnership with Fan Zhang, who is a research scientist in the lab of Shomara Chowdhury, a computational biologist and rheumatologist in our division. And I've also had help from Deepak Rao and other people who are part of the Accelerating Medicines Partnership, the NIH-FNIH consortium that has been looking at rheumatoid arthritis and lupus for the past several years. And with that, I'm happy to take questions.
Hey, thank you very much for a really, really interesting talk. And I see we've accumulated some questions, but I'm going to start with one of my own, if you don't mind. So Granzyme K is an active protease, it seems to me. Do the effects of it, for example, on augmenting cytokine effects, depend on its protease activity?
We think so, although we have not been able to show whether or not that's the case. There are no specific inhibitors for Granzyme K. There are only sort of very general protease inhibitors. It's almost easier to sort of try to identify the protein target of Granzyme K and then figure out if that, if that needs to be cleaved for it to be activated. And that's what we're looking at now. So we have, there are a couple of candidates in the literature for Granzyme K targets that we have not been able to verify, but we found some new ones that we're currently working on in the lab. So it's a work in progress.
Okay, thanks. So the first question, and I can read them and you answer. It's probably
that would be great.
Questions as well, but so the first question is from Wayne Tsuji, who asks, do CD8 positive Granzyme B positive and CD8 positive Granzyme K positive cells respond similarly to IL-15?
Yes. So we've stimulated cells with IL-12 and IL-15 together because it seems you need the little bit of an IL-12 boost to get IL-15 to do much of anything. And the Granzyme B positive cells and the Granzyme K positive B positive cells respond similarly to IL-12 and IL-15 stimulation. Interestingly, the cells that express Granzyme K alone do not respond. So there's a difference there where naive CD8 T cells and cells that make Granzyme K alone do not have permission to respond to cytokines. They don't have the ability to respond to cytokines, but the Granzyme B positive CD8 T cells and the Granzyme K positive B positive CD8 T cells, they do have the ability to respond to cytokines, which I think is going to be really important in a sort of a localized cytokine storm, such as rheumatoid arthritis synovium.
Okay, thanks. So next question is from Keith Elcon. He's asking two questions, but the first one is, what is the concentration of Granzyme K in synovial fluid?
That's a good question. We don't really know. So this is one of the challenges of working on such a relatively obscure protein. There are technically commercially available Granzyme K ELISAs, but they're very tricky and we haven't been able to get them to work. And so this is something we're actively looking at. We know that there's Granzyme B and Granzyme A in synovial fluid. That's been published before. We know that there's Granzyme K in serum, in plasma, both in healthy individuals and in sepsis and in rheumatoid arthritis, but we don't yet know what the level of Granzyme K is in synovial fluid. I would say that, I think that CD8 T cells, they're probably releasing it non-specifically potentially, but CD8 T cells are also likely forming some sort of synapse with target cells, right? So I think that there's going to be a local concentration of Granzyme K that's very important. And so knowing the diffuse, diluted Granzyme K concentration in synovial fluid might not be the whole story.
So Dr. Elcon's next question is there a Granzyme K knockout And is there any effect in it?
So a group, I believe it's in Australia, created a Granzyme K knockout mouse about 5 years ago. So before I started working on this project. And there are 2 papers published on this mouse. One of them from the primary group that made the mouse, they gave these mice every single infection they could think of, and they saw no phenotype. The other group that received these mice for an experiment, they were looking at chikungunya virus mediated arthritis. And they found that Granzyme K knockout mice had less inflammation in their joints than wild type mice in chikungunya mediated arthritis, or chikungunya virus associated arthritis, I should say. And so we're very curious to know what the effect of Granzyme K is, the Granzyme K knockout mice, if they have, to run some more traditional autoimmune arthritis models on those mice. So we're working on trying to get them. They're not commercially available, but we're trying to get our hands on some.
So the next question comes from Wayne Tsuji. It is, have you looked at IELs in celiac disease for Granzyme K positive versus B positive CD8 T cells?
No, we have not had the opportunity to do flow cytometry or anything like that on biopsies from gut. We have not looked at single cell RNA sequencing of celiac disease, but that's something that we should do. That would be very interesting to see, since that's a different, it's not an autoimmune disease per se in the way that IBD or rheumatoid arthritis is, and it's not an infection. So that'd be a different kind of scenario to look at. Yeah, we should definitely do that.
So I have one more question. I'm wondering if you look what Granzyme K might be doing to or what these cells, CD8 cells, double positive, what they might be doing to neutrophils. Do they affect neutrophils in some way?
Yeah, that is a good question, and I don't know the answer to that. We have not done, most of the samples that we use have been cryopreserved, and so most of the neutrophils, all the synovial fluid has gone through phycol processing, which removes most of the neutrophils, and I think the remaining neutrophils tend to lyse during the freeze-thaw process. Mm-hmm. And so we haven't gotten a chance to look at that specifically, but one could imagine that Granzyme K, I wouldn't be at all surprised if Granzyme K activates neutrophils, and that could, in theory, have ripple effects on the inflammation RA synovium. And so that'll be easier to look at in mouse models, for example. So we're hoping to do that.
So we have a question from Grant Hughes. Given the disease-specific differences in Granzyme B expression in viral infection and SLE versus other inflammatory conditions, what is known about type 1 and 3 interferons and Granzyme B induction? Yes,
so the regulation of Granzyme K and Granzyme B expression is something we're very interested in. We've been trying to figure out, the TCR repertoire data that I showed indicates that there's either recirculation or at least shared phenotypes between the blood and the synovial fluid pools of these Granzyme K positive CD8 T cells, but it doesn't explain like how they appear to begin with. How does a CD8 T cell wind up with this Granzyme K positive, Granzyme B positive phenotype and then stay there, right? And thereby accumulate in these inflamed tissues. We have tried various things, and nothing we've tried has induced Granzyme K positive, Granzyme B positive expression. If you stimulate CD8 T cells with through their TCRs, so with like CD3, CD28 beads, for example, within 3 days, the entire T cell population expresses only Granzyme B. A very sharp Granzyme B positive population, they look just like CTLs. So it's not a T cell mediated process. It's probably some sort of cytokine mix. We've looked at interferon alpha, we've looked at interferon gamma, we've looked at a whole bunch of other cytokines, and we see tiny little changes, but not the big accumulation of these double positive, Granzyme K positive, Granzyme B positive CD8 T cells that we want to see. And so we should look further into the interferon family. I think interferons for a long time were felt to not play a huge role in RA pathophysiology. I think that it's very difficult to detect interferon gamma protein And so levels were thought to be low, but now with RNA-seq, we see that interferon signatures, both type 1 and type 2, are very prominent in rheumatoid arthritis and in a number of other diseases, including, there was a lupus proteomic study that came out, right, a couple of years ago from Andrea Fava and others, and they found interferon gamma signatures among the cells in urine from people with lupus nephritis, which was a bit of a surprise, right? That was an interferon gamma signature that correlated with CD8 T cells. And so I think there's a lot about interferons that we don't understand, and looking further into the effect of interferon on both granzyme B expression and granzyme K expression is something we're planning to do.
So did you ever co-culture CD8s with synovial fibroblasts to see if it's some cell contact that would induce granzyme K expression?
We've done some of those experiments and we haven't seen a big effect, but I think that, the rheumatoid arthritis synovium is very complex. And so I think that you might need a combination of, cell-cell contact with maybe like some kind of integrins or, something like that, plus a cocktail of cytokines that have to be just right. And so our theory right now is that it's probably TCR independent, and then a mixture of cell-cell interactions and cytokines. And we're trying to figure out exactly what combination it is.
So the next question is sort of along the same lines, comes from Bobby Hahn. Do granzyme K positive CD8 T cells recognize specific antigens, and how do they interact with fibroblasts?
Yeah, so we don't yet know if these are antigen specific or not. So these single cell TCR repertoire studies have just become, financially possible thanks to 10x technology and so forth. And so as I'm part of the Accelerating Medicines Partnership, and as part of their current studies, they're doing TCR repertoire of a large number of synovial tissue and matched blood samples. And so we have a much larger TCR repertoire data set than the one that I showed a few slides ago. And I think that's going to be something that we'll be able to start seeing there. We're collaborating with a different group here at Brigham and Women's Hospital who are starting to look, their specialty is finding like the viral antigens of CD8 T cells in the context of viral infections and so forth. And so there's a postdoc there who's looking at the TCR sequences from these granzyme K positive CD8 T cells to see if she can determine the antigen specificity there. I think it's potentially very likely that these are viral CD8 T cells that were activated, in some past adenovirus infection or EBV or CMV or something like that. And then that gives them certain powers, right? So maybe the ability to respond to cytokines and things like that. And then they're recruited through their chemokine receptors to go to RA synovium. And then they get involved in this big inflammatory response in a totally antigen independent way. Like, I think that that's very possible. And that could be one way in which tolerance is broken. And so, I think in the next few years, we'll find out a lot more about the antigen specificity of these T cells. And I'm really looking forward to seeing what it is.
So would you expect that there's a relationship to citrullination?
I think there could be. I think so citrullination, a lot of that happens outside the cells, right? Where you have, the DNA and all the proteins on top of it. Is granzyme K also cutting these proteins and modifying the things that are stuck to the DNA? Like, is that one of the pieces that contributes to autoantibody production? I think that's possible. And I think it's going to take a little time to dig through all of this, but there are a lot of different possibilities. Mm-hmm. And how they interact with fibroblasts, we're not sure. Like, we have some potential receptor interactions that they might be doing. I think CD8 T cells, CD8 T cells are interesting because they sort of straddle the space between CD4 T cells and NK cells. So CD8 T cells express a lot of the same activating and inhibitory receptors that NK cells express, including, for example, NKG2D. And we know that MYC-A and MYC-B, which are two stress-induced ligands of NKG2D, are expressed by fibroblasts in rheumatoid arthritis synovium. And so it's possible that, there are other receptor ligand interactions that could be occurring here that are TCR independent. Mm-hmm.
All right. So that is the end of the written questions. So if in the next seconds somebody types something more, we can ask more. But in the meantime, I'd like to thank you very, very much for a great seminar. And I know many of us will continue the discussion with you in one-on-one settings. So thanks.
Yes, thank you so much.
Bye-bye.
Yes, thank you, Dr. Jonsson.